Okay, Zoom is behaving really weirdly. Well, just wait for the computer to wake up. I don't even know if anyone's going to join. People requested that I do Zoom, so I made Zoom. But 2 people said, we're on a noisy bus. We're not going to join any Zoom call. When you get a chance, congratulate our volleyball team for a remarkable win against Mount Vernon.
Yes, congratulations. Go volleyball. Thank you.
If you watch the highlights on Facebook, there are a couple of saves. People running almost into the bleachers to get the ball. Yeah. It was, I mean, I'd have just given up and said, okay, it's out, we're done. So that was impressive. Okay, what's going on with this? Are there any other quizzes out? All right. And don't forget to pick up your lab books. Other lab books, if you weren't able to pick them up on Friday, I will leave them in the natural sciences office. You can pick them up there. Very good. Let's have a listen to this little video about groups of enzymes called restriction endonucleases. Restriction endonucleases. Let's give a listen to this. Okay, stop. It's not. Okay, that's turned up. That's turned up. All right, let's try it again. Nope, sound's not working. Okay, that's on. Okay, now let's try it. Okay, it's there. It is. All right, there we go.
Restriction endonucleases are enzymes that cleave DNA into nucleotide sequences. The sequence recognized is often 4 to 6 nucleotides long. For example, the restriction endonuclease EcoR1 recognizes the sequence GAA-TTC. The nucleotides at one end of the recognition sequence are often complementary to those at the other end. The two strands of the DNA duplex have the same nucleotide sequence running in opposite directions for the length of the recognition sequence. Because the same recognition sequence occurs in both strands of the DNA duplex, the restriction endonuclease can bind to and cleave both strands of the DNA molecule. Because the bond cleaved is typically not positioned in the center of the recognition sequence, and the DNA strands are antiparallel, the cut sites are offset from each other. After cleavage, each DNA fragment has a single-stranded end that is a few nucleotides long. The single-stranded ends of the two fragments are complementary to each other. These single-stranded ends, sticky ends, can pair with each other. Once their ends have paired, the two fragments can be joined together by the enzyme DNA ligase, which reforms the phosphodiester bond of DNA. What makes restriction endonucleases so valuable is that any two fragments of DNA produced by the same restriction endonuclease can be joined together. Restriction endonucleases are fundamental tools in genetic engineering.
Come on, you. Behave. This is my blackboard for today. Since we're recording, this will end up on the recording. All right, so what that video talked about is the very basis of cloning. In the 1970s, there was a meeting between a venture capitalist and a scientist. And there, if you go to Genentech headquarters, there's something called Founders Park. And there's a sculpture of Herbert Boyer, the scientist, having a beer with a venture capitalist whose name escapes me right now. Anyway, the scientist is sitting there with a skeptical look on his face. His beer is hardly touched. And the venture capitalist is gesticulating wildly. His beer is almost completely gone. And that was the meeting that started the genetic engineering corporation, Genentech. Genentech makes a lot of things that you will administer in your careers. Insulin, that's made by recombinant DNA technology in recombinant bacteria or recombinant yeast. Those vectors were first made by Genentech, and then they were licensed to Eli Lilly to produce. So even though Eli Lilly produces it, it was Genentech that actually design the vectors that actually make the insulin. So in this regard, oh, that is really hard to see, isn't it? Let's do something that's a little easier to see. All right, better? Let's say this is a DNA molecule. Let's say this is DNA from a frog. Ribbit, ribbit, ribbit. Now, these enzymes that come from bacteria, called restriction endonucleases. Over there, buddy. Stay with me. It's a wrestling match with this thing. These were first found in bacteria. What they do in bacteria is the following. So let's say you have a bacterium, and that bacterium is infected by a virus. And that bacteria contains this viral DNA. So it injects that DNA into the bacterium. This DNA then has its genes expressed. Those gene products reprogram the cell to become a virus-producing factory. In order to protect itself from these invading viruses, these bacteria make these restriction endonuclease enzymes. These enzymes cut DNA at specific sequence motifs. So now, instead of this DNA being expressed, it'll encounter these enzymes and they'll cut DNA into small innocuous pieces. Now the bacterium will just gobble down those pieces. Bon appetit. And they're protected from the virus. So by harvesting these enzymes from bacteria, you can use them for genetic engineering purposes. So if you give these enzymes, say, to frog DNA, it'll cut frog DNA at very specific sequence motifs. Yeah, feel free to shut that door. When I came in this morning, the door was open because the janitor was cleaning the room, and she propped open the door, and that's why it's still open. And as the video talked about, this DNA will have so-called sticky ends. The DNA will have some overlaps like this. And these ends can re-anneal to each other if need be. Now let's suppose you use the same restriction endonuclease to cut plasmid DNA from a bacterium. These two sets of sticky ends, the sequence on the ends here is the same. So the sequence here is complementary to this sequence here. This sequence here is complementary to this sequence here. So what you should be able to do then is take this DNA that was cut with the same restriction endonuclease, put it in a test tube, then put it at very low temperature with high salt in the presence of this DNA, this repair enzyme in cells called DNA ligase. DNA ligase, you'll remember, plays a role in DNA replication. And you can get then you can then get a molecule that is part frog, part bacterium, can't you? That's exactly the first recombinant DNA experiment that was done by Stanley Cohen and Herbert Boyer in 1976. It was published in Proceedings of the National Academy of Sciences. This has revolutionized science. It's revolutionized biology. It's revolutionized medicine in particular. So now you can take this plasmid, you could stick it back into a bacterium. The bacterium will make bucket loads of this DNA and it'll express that gene. And you can get this bacterium now to become a factory to express this gene, be it insulin, be it human growth hormone, be it cytokines, be it whatever. And so this recombinant DNA technology essentially uses this same strategy. Some of the players essentially are a little fancier and what have you, but the strategy is still basically the same. You use restriction endonucleases, you cut two DNA molecules from different species with the same enzyme, you ligate them together, you put them in a bacterium, you screen them for the one that will make the gene that you're interested in, and then you use that strain to make lots of whatever you wanted to make, market it for an exorbitant price, and retire before you're at the age of 40. Or so the story goes. So this shows you essentially how genetic engineering uses bacteria and uses enzymes that were first isolated from bacteria. Enzymes that were originally just a curiosity that bacteria used to protect themselves from being infected by viruses. Now, interestingly, the video told you the enzyme that they talked about was EcoR1. E comes from Escherichia. Co comes from coli. R1 comes from strain R1. These are named after the strains they come from. So for example, there is a enzyme that's called HindD3. HindD3 comes from Haemophilus influenzae strain D3. Or there's another one that's called BAM-H1. That comes from Bacillus amylophilus strain H1. Blah, 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 so on and so forth. If you go to the New England Biolabs catalog, there's hundreds of these enzymes. They cut at hundreds of different sequence motifs. So you can find enzymes that cut at 6 base motifs, 4 base motifs, enzymes that cut with 3' overhangs, enzymes that cut with 5' overhangs, enzymes that are blunt end cutters, 8 cutters, things that cut at 8 sequence motifs. The sky's the limit. You have just about every enzyme you could ask for, essentially. Okay, computer. Gonna behave? Every time I make this computer do Zoom, it gets very sluggish. Come on, you. There you go. So the enzymes sometimes cut. The video made it sound like all restriction enzymes cut at these sequences that say the same thing in one direction as they do at the other. Those types of sequences are called palindromes. Palindromes. So a very famous palindrome to illustrate how palindromes work. If we were to say, Madam, in Eden, I'm Adam. Now read it the other way. Madam, in Eden, I'm Adam. Reads the same way in one direction as it does in the other. Stan Arts, a professor of mine at UC Davis, he collected these in 15 different languages. The oldest one is in Greek, and it was found at a 4th century baptismal font. And it's a Greek palindrome. The longest palindrome is the Finnish word for a dealer in lie. Don't ask me to try and say it because I don't remember it and I wouldn't say it right anyway. But he had pages and pages of these. But in molecular biology, if a sequence runs the same way in the 5' to 3' end as it does in the 3' to 5' end, it's a palindrome. And many of these restriction endonucleases cut at palindromic sequences. The fragment that you get is called a restriction fragment. Those fragments will vary because the sequence motif appears in the genome according to chance. And you can do calculations essentially that will tell you how often you'd expect a certain restriction endonuclease site to appear in DNA. Let's say, for example, your restriction endonuclease cuts at a 4-base motif. How many bases are there in DNA? Yeah, good, Natalie. 4, yeah. That's not a trick question. Adenine, guanine, cytosine, thymine. 4. So how often is a 4-base motif going to occur? 4 to the 4th power. That's equal to 256. So you should expect to see, if your enzyme cuts at a 4-base motif, your average restriction fragment should be 256 base pairs long. That's your average. Some will be a lot bigger, some will be a lot shorter. But if you do a curve of how often they occur and the size of the fragment, you should expect to get a bell curve, and the mean of this should be 256. And guess what? If you cut bacterial DNA with a 4-cutter, like Sal3A, that's exactly what you find. If they cut it a 6-base motif, 4 to the 6th power. I'll let you figure out what that one is. Your science notation calculator can do that very quickly. And then there's some that are 8-cutters. When you're playing with a calculator some night, if you're doing statistics, just calculate that and it'll tell you how often you would expect to find these particular lengths. Now, because sequences will vary between different strains of bacteria, when you cut them with different restriction endonucleases, you'll get restriction fragments that will vary in size. We call those differences in size of the restriction fragments. We call those restriction fragment length polymorphisms. Restriction fragment length polymorphisms. Restriction fragment length polymorphisms are useful when it comes to human DNA because certain genetic diseases correlate with the occurrence of certain restriction fragment length polymorphisms. So RFLPs, restriction fragment length polymorphisms, can be remarkably useful as ways to detect the presence of certain genetic diseases. So this illustrates in pictorial fashion the use of restriction endonucleases to cut various types of DNA and then annealing it to plasmid DNA, putting this back in the bacterium and have the bacterium make lots of it. Here are some of the recognition motifs recognized by some of these different enzymes Here's one that cuts right in between and produces blunt ends. Those blunt-ended cutters can be very useful for cloning PCR products and things like that. And then ligase, of course, is necessary to take those hybrid molecules and to seal up the nicks in between the backbones of the DNA molecule. Now, what if you wanted to clone something that comes from messenger RNA? You need a way to make DNA from RNA. Fortunately, reverse transcriptase is available to do that. And there are commercially available reverse transcriptase lots that you can buy from lots of different companies. Isolating messenger RNA is relatively straightforward. There are kits you can buy that will isolate messenger RNA from eukaryotic cells. So messenger RNA I'll be a good citizen and do this on my makeshift blackboard over here. So this is messenger RNA, say from, oh, I don't know, liver cells, let's say. In eukaryotes, messenger RNAs has a string of adenines at the end of it. I'm sorry? Yes, you are supposed to see that. Oh, doggone it. Messenger RNA has I know what's wrong. The tedium of using Zoom is remarkable. So messenger RNA in eukaryotes has what at the 3' end has this thing called a poly A tail. After transcription terminates, there's an enzyme that adds about 250 adenines to the end of the tail. Bacteria don't do that, just eukaryotic cells. You can use this as a handle to isolate messenger RNA from eukaryotic cells really easily. You take a bead and you attach to that bead and you attach to that bead a bunch, something with a bunch of T's linked to it. You incubate the RNA with these beads, and this will anneal to that. Because this anneals to that, you isolate messenger RNA. Most of the RNA inside the cell is ribosomal RNA by quantity. Probably 60 to 70% of the RNA in the cell is ribosomal RNA. So isolating your messenger RNA can be hard to do, but using these oligo-DT beads, as they're called, you can isolate this really well. These beads now are magnetized. So you can literally throw your RNA in a little test tube, put a magnet up to it, dump everything out, wash it, dump everything out, and then take the magnet off, and then dissociate this, and you have pure messenger RNA. It'll take you 2 hours to isolate messenger RNA. So that's much easier to do. In fact, my students did this last fall. last spring. It's remarkably easy to do. Okay, Jeopardy thing plays. Come on, computer. So if you make a copy of messenger RNA and then clone that, you have made what's called copy or complementary DNA, which is abbreviated cDNA. So if you see the term cDNA, it means it was made from messenger RNA. Polymerase chain reaction is a very, very heavily used technique to detect exquisitely small amounts of nucleic acid. Many of the diagnostic tests that will be ordered, whose results you will interpret, or tests that you yourself will order, are so-called nucleic acid amplification tests. These nucleic acid amplification tests are polymerase chain reaction tests for your pathogen. We have a video that shows you how polymerase chain reaction works. Let's go ahead and watch that. I have to click everything twice. Boy, that gets old fast. Okay, let's go back to the buzz.
The polymerase chain reaction is a method for making many copies of a specific segment of DNA, starting with a very small amount. This technique can be used to identify specific microorganisms from a small amount of DNA and to identify persons involved in crimes from DNA on cigarettes or in a single hair follicle. The DNA to be amplified is mixed with deoxyribonucleotides, a thermal stable DNA polymerase called Taq polymerase, and DNA primers. The DNA primers hybridize to the ends of the chain to be amplified and provide a starting point for the Taq polymerase. The mixture is heated to break the hydrogen bonds in the DNA, forming single-stranded molecules. The mixture is then cooled sufficiently to allow the DNA primers to anneal to each end of the segment to be copied. Taq polymerase then synthesizes the complementary strand of DNA using the primer as the starting point. The temperature is raised again to separate the DNA strands and then lowered sufficiently to allow the primers to attach. Taq polymerase now synthesizes another set of new complementary strands. This process is repeated until enough DNA has been produced to be identified or used for further research. After 21 cycles, one molecule of DNA can be amplified to over 1 million copies. This amount of amplification can be achieved by running the reaction overnight in a thermal cycler, an instrument that automatically raises and lowers the temperature at appropriate time intervals.
Yeah, that's better. So that's polymerase chain reaction. You start out with a very, very small amount of DNA. You then amplify it using this programmed cycle of annealing, extending, denaturing, annealing, extending, denaturing, annealing, extending, denaturing, annealing, extending, denaturing, so on and so forth. By using primers that can hybridize to your DNA in question, you can make enormous amounts of your DNA that you can actually analyze. So you started from really, really small amounts of DNA, and now you have enough that you can analyze. It's especially useful for detecting if something is actually there. So let's say, for example, a lady comes into your clinic and she has what you think is a STI, but you don't really want to wait for a culture. You want to know if she has a particular STI agent, so you order a polymerase chain reaction. The polymerase chain reaction can take small amounts of DNA just from a vaginal swab or something like that. You can make lots and lots and lots of that DNA. That tells you that pathogen is there. Because that pathogen is there, you know it's probably susceptible to these antibiotics. You can treat her with these antibiotics, and you don't have to wait a day or two for a culture or something like that. Yeah, if I click everything twice, maybe it'll happen. All right, so that's polymerase chain reaction. The important element in polymerase chain reaction is this heat-resistant DNA polymerase that is isolated from an aquatic bacterium that lives in hot springs. The organism is called Thermus aquaticus, so the enzyme gets called Taq polymerase after Thermus aquaticus. There are several heat-resistant DNA polymerases now that are available commercially, some of which are not derived from Thermus aquaticus. There's an enzyme now called the Vent polymerase that comes from a completely different organism. All of these, however, are able to work at these high temperatures. So when the DNA gets denatured, you don't kill the enzyme. And this repeated cycle of annealing, extending, or polymerizing, denaturing, and what have you, you start out with very small amounts of DNA, and then you make lots of it. This is the exact test that you have during a COVID test. So you get brained essentially with a swab, goes way up in your nasal cavity, and it doesn't feel very nice. And then that swab, essentially, you give the material in the swab, the nucleotides, the enzyme, the primers, so on and so forth. Since we know the sequence of SARS-CoV-2, you use primers specific for it, and then you amplify it. And then if that's detectable, it says you have virus in your nasal pharynx. How do you tell if that DNA is there? You use a technique called gel electrophoresis. You put the fragments into an agarose gel. DNA is negatively charged. So if you put a charge across that gel and put it in a solution of electrolytes, so it can carry charge, the DNA will migrate to the positive pole. Agarose gels have certain molecular pore sizes. So small DNA fragments will move through the gel very quickly. Large DNA fragments will move much more slowly. So you separate your fragments on the basis of size. And you'll get a banding pattern that looks like this. That's a very pretty gel. My gels don't necessarily look that way, but that's a very pretty one. And this is just an example, essentially, of what these gels will look like. You can stain these DNA fragments using either fluorescent stains, like ethidium bromide. You can also use visible stains. There's a stain you can use called visible blue. What? Say that again. Oh, okay, and you're having nightmares. Oh, okay. All right, well, wake up. All right, so these sorts of analyses then tell you if the fragments are there, and they also tell you what size the fragments are. So gel electrophoresis with PCR is remarkably powerful. We also have small primers that can amplify pieces from just about every chromosome in your body. Those fragments that the PCR reaction will amplify vary from person to person. Because of that, you can use PCR to get a genetic fingerprint of your DNA. This is essentially how police departments know if material left at a crime scene came from you or came from somebody else. They can essentially specify with high precision where that DNA came from. And so if blood or hair or some other bodily fluid is left at a crime scene, this is how the police labs can determine if it came from you or someone else. This is also how paternity tests are done as well. A similar strategy can be used to sequence DNA. And I don't want to read you the riot act on how DNA is sequenced, but very simply, you use modified bases. Those modified bases, when they're put in, you can then use a laser to excite those dyes. And because each base is labeled by a different dye, it will fluoresce at a different frequency. And the photodetectors will then tell you which dye got excited. And based on that, the computer will tell you essentially the sequence of your DNA. So this is called high throughput sequencing. High throughput sequencing machines can sequence enormously long fragments. Now with automated sequencing machines, they can sequence many, many long fragments at a time. And then the computer can detect if there's sequence that overlaps. If there's sequence that overlaps, the computer can then put it together. And this is essentially how you can sequence whole genomes essentially in an afternoon. These machines are called 454 sequencing machines. 454 sequencing machines. You could sequence an entire human genome in an afternoon with these machines. Think about that again, 3 billion bases of DNA. You can sequence that in an afternoon. And you can sequence an entire human genome for a fraction of the cost that you could just 10 years ago. This technology just keeps getting better and better and better and better and better. And when we do genetics next semester, there'll be some genetic diseases for which they're very good tests. However, for others, well, you need to sequence a genome. There will come a time when this will probably get so cheap where you can probably just say to a family, oh, to find out, let's just sequence your kid's genome. Because it'll be in the hundreds of dollars and not thousands of dollars. And that's something insurance now will easily pay for, because that will help them assess the risk when it comes to insuring your kid. So genomics is the study of the genes in an organism. A genome, that's the collective amount of DNA you have in your cells and all the genes that are in your cells. The protein study of proteinomics is a study of the proteins that the genome encodes. Metagenomics looks at the sum total of the genomes of the organisms that occupy a particular ecological niche. So if we looked at all the genomes of the bacteria that colonize your skin, that would be a metagenomic study. Or your GI tract, or your scalp, or your between your toes, or whatever. That'd be a metagenomic study. Metabolomics essentially looks at the complete array of small metabolites, small chemicals present in a cell or present outside a cell at any time. So you get a snapshot, if you will, of the physiological state of the cell, its end products, and so on and so forth. So what we've been talking about, recombinant DNA technology can allow cells to make enzymes and hormones and even vaccines. The hepatitis B vaccine you received is a genetically engineered vaccine. So are several others that you received as well. The DTaP, the toxoid part of DTaP, that's genetically engineered. The tetanus booster you received, that's genetically engineered. Lots of the things you receive are genetically engineered. Any sort of protein that you get. So for example, there's a drug, there are commercials for it. If you are in the gym and you watch ESPN or any of those other tasteless commercials that come on in the middle of ESPN, there's a commercial for Neupogen. It's for cancer patients to raise their white cells during chemotherapy. That is colony stimulating factor. It's a type of colony stimulating factor. That's a small protein that's made in genetically engineered yeast. And then just about all the hormones that you give are usually made by genetically engineered organisms, with the exception of steroids. Steroids are usually made chemically. So when we talk about gene cloning, it means that we take a gene from a host organism, we isolate it in a so-called cloning vector. That vector is usually a plasmid, but it can also be a virus. The vector then takes the piece of DNA from the organism of interest. You put it into a bacterium, you make lots of it, and then you express the gene of interest. And here's the strategy that we talked about before at the beginning of class. I won't go over this again because, yeah. So this has led to a whole new field called synthetic biology. Now there's the ability to make synthetic DNA molecules. This process used to be remarkably tedious and expensive. Now it's been automated. Since it's been automated and the kinks have been worked out, the prices come way down, and now it's possible to make DNA molecules that are even longer. So in 2010, Craig Ventnor made a molecule, DNA molecule, of the bacterium Mycoplasma parasitica, and he put that into mycoplasma cells, and it worked. So this led to this whole new field of synthetic biology. Synthetic biology essentially looks at making missing molecules that are needed in order to fix a certain genetic disease, or to assemble even more complicated molecules that you can give to people who are missing something, or if something is broken, you can supply it, what have you, because of the ability to make DNA molecules and synthetic proteins in the test tube and then give them to patients. That's created new avenues of research and therapeutics as well. And now there's this technique called CRISPR. CRISPR allows scientists to alter genomes in sequence-specific ways. When CRISPR was first discovered by Jennifer Doudna, for which she won the Nobel Prize, it was very dirty. When it was used, there were lots of off-target modifications to the genome. There are still off-target modifications to the genome. with CRISPR. The number of off-target modifications has gone way down. It's cleaner, but there's talk now of using this to actually treat genetic diseases in embryos. People have gotten this to work in mice. Now, just because you get something to work in mice doesn't mean it'll work in humans. When gene therapy worked extremely well in mice, and then it was tried in humans, we got the Jesse Gelsinger disaster at the beginning of the 21st century. That put gene therapy on hold for the next 20 years. So just because it's worked in mice doesn't mean it'll work necessarily in humans. So I personally think we need to step back and say, let's pump the brakes. But people want to charge forward, seemingly forgetting the disaster that occurred with gene therapy. Furthermore, I think when you start modifying embryos, where's it going to stop? I'm all for curing genetic diseases, but do you think it's going to stop there? What about genes to make people more muscular? What about genes to make people smarter? Now let's really get to where it lives. What about genes to change people's skin color? Do you think it's really going to stop there? I'm a strong believer in human depravity. I think we need to pump the brakes on this technology big time. Yes, Brooke. Jesse Gelsinger was a patient in a gene therapy trial at the University of Pennsylvania. I was actually there as a postdoc when it happened. So he had a liver disease that was inherited. With medication, his disease was well tolerated. He was living a normal life. He volunteered for the gene therapy because he believed in the technology. He said, there are people with the same disease I have whose disease is not as mild, whose disease is not controlled. So he was given the vector and he experienced a remarkably huge immune response against the gene therapy vector. And he essentially experienced a cytokine storm and it killed him. He was 18 years old. This was completely not conceived of. This shocked everybody because it shouldn't have happened. He was given a test dose of the virus. He didn't react to it, but what that probably did was sensitize the system to the virus. And so when he was given a much larger dose of it, he got a cytokine storm because it was made to infect his liver cells. And so now he had this enormous immune response against his liver. His liver function tanked, and then everything else tanked. His organs shut down, and he died quickly. So his dad, of course, sued the University of Pennsylvania. They deserved it because they were warned by people, you're going too fast. Just because this works so well in mice doesn't mean it's going to work so well in humans. We had the same sort of thing with embryonic stem cells. Oh, look, it works so well in mice. It's going to work great in humans. Survey says, no, 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 no. Which part don't you understand, the N or the O? No. And so people are saying the same thing. Oh, look, CRISPR works so well in mice. Haven't we learned how slowly we learn? So CRISPR, again, comes from bacteria. It's a system from bacteria to protect them from invading viruses. And I think this is remarkable technology that holds promise, but I think it's going to need to be very, very, very carefully curtailed. Just because I think when you start getting profit margins combined with the temptation for perfect babies, I think that's a perfect storm. And who's going to pay the price? A lot of unborn children are going to pay the price. Okay. Restriction endonucleases. Do they make cuts to DNA at palindromic sequences? Not all of them do, but most of them do. So I call that a, yeah. Leave sticky ends at the ends of DNA cuts. Yeah, a lot of them do. Not all of them do, but a lot of them do. So I call that a, yeah, probably. are used by bacteria to protect against invading bacteriophage. Yep, that's how they were discovered. Are important introducing a gene into a vector in genetic engineering. That's what we use them for. So, are all these choices correct? You bet your sandals. All right, are there any questions about anything from chapter 8 before we take our break? Nope. Okay, we will take a break, start chapter 9. Doesn't look like anybody joined us for Zoom today, but that's okay. So in 5 minutes, we will resume. Yours is, that's Haley's one. It's Jessica's not here. Marie's not here. Oh, Hannah. Maddie, can you put that right, that computer there? Oh, McKenna. Brianna. Can you hand that to Brianna, please?
What will the chapter 9 exam be due online?
Probably next week sometime. Macy's not here. Emily's not here. Oh, Natalie. Oh, Sydney. Yeah. It'll be due next week sometime. Boy, they turn the heat on in here with a vengeance. November. Second Friday in November.
The next one.
Yeah. Yeah, I'm pretty sure it's second Friday in November. Yeah. Yeah. And after the recording snafu we had with my seminar, Mrs. Schaefer called IT and unloaded on them, shall we say. We had that happen multiple times last year. And so she said, look, you guys hired a full-time person whom you pay to do this. Tell them to get their body parts there and record our seminars instead of leaving it to part-time people who may not know what they're doing as well as the full-time person does. And so the full-time person was there last time. We didn't have the snafu we had before. And so hopefully these will be recorded instead of like what happened with my seminar. Because they came to me after my seminar and they said, well, could you essentially redo it and record it on your own time? To which I said, no. Not that I don't want to, it's just that I'm just too darn busy. So, all right, it's time to resume. So let us resume, shall we say. So we're now going to embark on a discussion of how to make sure that you control microbe numbers. This is going to be a huge part of what you will do as nurses. There's something that you'll have to respect a great deal when you're working, this thing called sterile fields. So let's say, for example, you're going to put in a Foley catheter. That Foley catheter comes in a package. That package has been sterilized usually by gamma radiation. The second you open it up, you're exposing it to the air. Now, hospital air is very heavily filtered using HEPA filter systems. However, if you touch that Foley catheter with your bare hands, you've now contaminated it. So once you open it, that's a sterile field. You also need to make sure wherever you lay that Foley catheter, the part that you're going to insert into the patient needs to always be sterile. Usually that Foley catheter will have within it a pair of gloves. You don the gloves. Once you don the gloves, you've donned sterile gloves. You don't don the gloves and then scratch your head because you've now violated that sterile field. If you violate a sterile field, you have to take the whole thing, wrap it up and throw it away and start again. That gets expensive for the hospital. So you want to make sure that you respect sterile fields. However, the human body can be a very germ-ridden thing. So how do you deal with this germ-ridden thing? You use chemicals that can either reduce or completely get rid of microbes. So you want to either reduce microbe numbers or you want to completely get rid of them. So sterilization simply means you get rid of every stinking microbe that's around. When we put things in the autoclave, we sterilize them. There are no more microbes in this liquid. And sterilization can take place using heat. It can take place using certain chemicals like propylium oxide or something like that. Or you can use ionizing radiation or ultraviolet radiation or something like that. So sterilization, when you open that trach or that Foley catheter or that IV set or something like that, or the needles that you're going to use, those are sterilized. There's no microbes in them at all. When you use them, you need to remember that and treat them as such. Disinfection. With disinfection, you're reducing microbe numbers. You're reducing microbe numbers to the point where you've reduced all the harmful microbes. So for example, when you come into the lab and you spray the surfaces down and you wipe it down, are you sterilizing the surface? No, you're disinfecting it. You're knocking those numbers down to the point where any potential pathogens have been reduced. So you're destroying vegetative pathogens, but you're not destroying bacterial endospores. You're removing harmful products and what have you. Decontamination or sanitization. Decontamination or sanitization. This is essentially what is used in your dishwasher. Your dishwasher goes through the wash cycle. Then there's usually a light that lights up, it says Sani. That's not the name of the repair person. That's short for sanitization. Now the dishwasher essentially is going to heat the utensils. By heating the utensils, it's going to destroy any bacteria that could potentially get you sick. So let's say, for example, you open your drawer where you keep your forks, knives, and everything else, your cutlery and utensils, and scurry, 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 you see a cockroach. Yes, Caitlin, they are disgusting. I worked with them once in the laboratory. They're still disgusting. They're quite easy to grow in the laboratory because they'll eat anything, but disgusting little creatures. I still don't like them. Now, are you going to eat with those forks and knives? Okay, can we all agree the answer is no? Don't you look at me as if saying, what's the right answer? Okay, the answer is no, okay? Roaches are remarkably good mechanical vectors for disease. They're covered with bristles. Those bristles grab microbes, and then as they walk across stuff, they leave microbes wherever they go. So they're really good mechanical vectors. And what sort of things do they crawl through? Fecal material, dead critters, things like that. They're really good at picking it up and they're really good at dropping it off. So the answer is a resounding no. Thank you. No. Same thing with houseflies. Houseflies walk across your food, your potato chip. You can eat that potato chip? Let's hear it. Houseflies are also extremely good mechanical vectors of disease. If you've ever looked at a housefly under intense magnification, they're covered from stem to stern with bristles. Those bristles are mechanoreceptors that they use to detect air currents and things like that. But those bristles also are very sticky. They're charged. So they attract lots of things. One of those things being bacteria. Also, the way they eat, they also do a really good job of taking up bacteria with their mouth parts as well. You've probably heard they vomit on things to eat. That's actually not true. That's a myth. That's a myth. Anyway, so you put your utensils in the dishwasher. The dishwasher uses detergent. That is a disinfectant. And they use heat. That's going to decontaminate and sanitize. Does it kill all microbes? No, but it reduces microbe numbers to a safe level. And yes, there is a safe level of microbes. Antiseptics. Antiseptics. Have you volunteered at blood banks or you have to, okay, what do you do before they come in, the donor? What do you do before you okay, you disinfect their arm. What do you use? So you use an alcohol swab first, and then you use the providone iodine solution that you can always tell someone's given blood, their arm's kind of yellowish. That's the iodine solution. That is an antiseptic. You are killing microbes on the skin. There's a group of organisms you want to kill. You can't take their arm and put it in the autoclave, can you? No, you can't take their arm and put it in your dishwasher. Oh, here, there, here. No, it doesn't work that way, does it? It needs to stay attached. So you use chemical agents that kill that small group of organisms on the skin. So you scrub the skin. That scrubbing creates friction with the chemicals. So use alcohol. If you do any surgical nursing, you'll be involved with this as well. That's antiseptics. Reducing microbe numbers on the skin. Reducing microbe numbers on the skin. So you can put in a PICC line or an IV or something like that. Yes, that gets rid of everything. Everything. I'm a Star Trek fan. Star Trek, the old Star Trek, there was this probe Nomad, and Nomad talked about sterilize. Sterilize, kept saying sterilize. I have just sterilized, return to source and sterilize. And they finally figured out it needs to go back to Earth and sterilize the Earth because the Earth is infested with people. So by sterilize the Earth meant kill all the people on the Earth. Yeah, so that's the old Star Trek. Your book gives this table, this little picture. This is a really useful table. So if you ever get stuck with this, just take this table. It's color coded. It's really well. Each one of these that say sterilize, they kill everything. So there are physical agents that will sterilize, chemical agents that will sterilize, mechanical filtration type liquid methods that will also sterilize. Disinfection is in the powder blue. Antisepsis, this is nice because it's the same color as the iodine solution. I think that's kind of cool. And then decontamination is over here kind of in the ochre yellow that you see there. All right, so let's talk about some of these techniques, some of these methods. We're going to do a lab next week that will involve sterilization and testing the ability of different agents to kill different types of microbes. So some microbes have relative resistance to certain types of sterilization. Also, the greater the number of microbes, the greater their resistance to sterilization. So if you take some soil, for example, and you boil soil, microbes from soil very often will clump together when you boil them. So you kill the cells on the outside, then you kill the next shell of cells, then you kill the next shell of cells, then you kill the next shell of cells. The cells at the very center are the hardest to kill. And after you boil them once, usually those microbes on the inside usually will still survive. This is also the case when you cook certain types of foods. That's why with certain types of foods, you have to cook them twice. You cook them before you put the lid on, then after they're vacuum sealed, you cook them again to make sure that that twice heating, you've knocked them down to where that food is absolutely microbe free by the time the consumer opens the can. But the goal of sterilization is to kill all microbes within the sample. This shows you the relative resistance of the different organisms. At the bottom, we have enveloped viruses. Envelopes are fairly easy to disrupt. Detergents will disrupt those. That's why soap, washing your hands with soap, can go a long way towards preventing the transmission of certain diseases. Then we have gram-positive bacteria, non-enveloped viruses, fungi and fungal spores, a lot of your gram-negatives, protozoans, protozoan cysts. We might put also in here cyst eggs from certain worms. Some eggs from certain worms can be very, very resistant, and they can contaminate certain foods under certain conditions. That's why we need to cook our food. Staph and pseudomonads are very tough. Mycobacteria are ridiculously resistant, and then at the top, we have bacterial endospores, and then at the very top, of course, we have prions, which is just naked proteins and are very hard to destroy. This shows you essentially the resistance of endospores versus vegetative forms. So heat, endospores are 1.5 times more resistant to heat than vegetative cells. For ionizing radiation, endospores are 4 times more resistant than vegetative cells. Sterilizing gas like ethylene oxide, endospores are roughly 2 times more resistant than vegetative cells. And when it comes to sporicidal liquids like glutaraldehyde or something like that, endospores are 18 times more resistant. What do these data tell you? They're much harder to kill. And if you are trying to sterilize something that has been contaminated with soil or other sources of endospores, and animals, many organisms associated with animals, animal products, and animal exposures can also be sources of endospores. You need to take into account endospores, and you need to subject that sample to modes of sterilization that will also kill endospores. Yes. Sterilization also gets rid of prions, yes. Yeah, sterilization essentially is a scorched earth technique. That's why sterilization, when it comes to medicine, works for products, but not for people. You can never really sterilize the human body, but you have to account for microbes. And that's why surgeons, when you do surgery, if you're dealing with areas that are microbe rich, you knock down the number of pathogens. And by knocking down the number of pathogens, even if you get some microbes in the body during surgery, those microbes in the body, they'll be such small numbers and they won't be pathogenic. They're of little consequence. Products, however, can be sterilized, and they must be sterilized before you introduce them into the body. So any sort of joint implants, any sort of catheters, any sort of lines, central lines, PICC lines, and so on and so forth. All right, so sterilization and disinfection types of processes. When we talk about things that are homicidal, so homicide, homicidal, side means to destroy. So bactericides, virucides, fungicides, sporicides, that means they destroy, they kill them. Germicides means they kill microbes. If, however, they use the term stat or static, it means they inhibit the growth of. So fungistatic organisms inhibit the growth of fungi, they don't kill fungi. Bacteriostatic agents inhibit the growth of bacteria, but they don't necessarily kill them. That'll be important when we look at antibiotics because some are bactericidal, some are bacteriostatic. Sepsis. Sepsis, that's a term you learned in pathophys. It means that you have the growth of microbes in the blood and in other tissues as well. Asepsis and aseptic techniques prevent the introduction of microbes into the body when you're subjecting someone to some sort of procedure. You're trying to prevent, remember this other term from pathophys, iatrogenic. Remember that term from path? I know it's been a while. Iatrogenic, an iatrogenic condition is a condition caused by the treatment. Does that ring a bell now? You guys are shaking your heads. That's not a term you came across. Okay, some of you are saying, yeah, I heard it, but I promptly forgot it after pathophys. Iatrogenic, it means that the treatment caused the condition. The treatment caused the condition. So for example, some types of anti-cancer drugs can cause mutations in cells, and they can increase your risk of cancers. So you have, say, for example, lung cancer. You beat the lung cancer, but you were treated with anti-cancer drugs, and then 20 years later, you now have leukemia. That leukemia is probably an iatrogenic leukemia that was caused by the anti-cancer drugs you were given for the lung cancer. Those drugs, however, bought you another 20 years. So antiseptics are used on the skin. On the skin. In dental procedures, they can be used on the gums. Because gums are loaded with microbes, particularly when you go in between, when your dentist takes or the hygienist takes that periodontal probe, and well, the majority of dental hygienists are she, so I'm going to say she. She's looking essentially at the numbers on the probe to see if your gums are receded. And it's a 4, looking for 4, 3, 2, 1, something like that. That surface, if you need any sort of work, needs to be cleaned before you can have any dental work. So antiseptics are used in that case. Hand soap is a type of antiseptic. When you guys do hand hygiene, when you come into the room before you deal with a patient, and you have that specific way you're supposed to wash hands to get the parts that people normally miss, thumbs, edges, in between the finger pits and so on and so forth, what have you. You are exercising antisepsis. Okay, we talked about static and cytal. Any questions on that? Go on. So when we talk about critical medical devices, if they come into contact with sterile tissues, they're critical medical devices. They must be sterilized before use. Semi-critical devices will come into contact with mucosal membranes. For example, G-tubes. G-tubes come into contact with mucosal membranes. Are mucosal membranes sterile? No. They must receive high-level disinfection. They'll usually be sterilized. When you take a G-tube out of its package, it's sterilized. And you insert it and so on and so forth, and they're disinfected. So high-level disinfection, dentures would be examples of semi-critical devices. Non-critical devices, they only make contact with the skin. They require low-level disinfection unless they've been contaminated by body fluids. Good example of this would be EKG electrodes. Do you reuse EKG electrodes? Nope, they're made disposable for a reason. You pitch them. And those electrodes, again, they're usually sterilized with gamma radiation. They're usually sterile, but they don't need to be. If you're in the ER and you have a trauma patient and they're bleeding and you need to get a quick EKG, or paddles, if you're using paddles, do those paddles need to be sterile before you use them? No, they need to be disinfected because they're just going on the skin. If someone's bleeding and you get blood on them, now you need to do high level disinfection in case like that. Okay, so sterilization, there's no one size fits all very often. Sometimes you're going to be working with compounds. For example, plastics. You can't put plastics in the autoclave. Why? They'll melt. Now, there are some plastics that are heat resistant. Really, really well polymerized polyurethane is autoclave resistant. Other types of plastics, you put those plastics in the autoclave, you'll have a pool of molten plastic. So those cannot undergo sterilization in that method. You must sterilize them using other methods. So you have to consider cost. Autoclaves, fortunately, are pretty cheap. Effectiveness, and then method of disposal as well. So those are all considerations. So what does it mean when a microbe is dead? When it's dead, it can no longer grow or divide or carry out metabolism. It can neither grow nor divide nor carry out metabolism. The cell might still be there, but it's so irreparably damaged that it just can't carry on the processes of life. When you have a population of microbes, death is not instantaneous. Microbial death is usually logarithmic. So if you have, say, 1 million microbes, you'll reduce microbes, given the way you're trying to sterilize them, by factors of 10 over periods of time. When eventually you get to the point where theoretically there's only 1 microbe left, and if you keep sterilizing then, you reduce by another factor of 10, all those microbes are dead. So, for example, here's a good example I think that will hit home. When you're at home, you're going to go home for Thanksgiving, and you say, oh, here, I'll help do the dishes. And you grab the sponge, and you squeeze the sponge, and this fluid comes out, and it's a funny colored fluid. And you look at it and you go, yeah, I don't think I should use this sponge. Oh, I know, I'll put some soap on it. That soap will kill all the microbes. Is that good microbiological thinking? No. There's a good chance microbes have been growing in that sponge for quite a while. You put soap in it, you're going to start to kill the microbes like this over a period of time. So while you're scrubbing with them, you're actually putting microbes on those pots and pans. Now, you're probably washing it off with hot water, but you're better off probably pitching that sponge and getting another one and using that sponge in that case. So you never just kill microbes like that. You kill them in a logarithmic manner. You kill them in orders of 10. So 10 to the 6th, then you reduce them 10 there. Now there's 10 to the 5th. Now there's 10 to, so million, 100,000, 10,000, 1,000, 100, 10, 1, zip over a period of time. How long does it take? Depends on your starting numbers, and it depends upon the method you're using, and it depends upon the organism, how resistant the organism is to killing. Active cells will die more quickly. Cells that are in stationary phase are harder to kill. There's a unit of time that we used in the food industry. It's also used in medicine. It's called a D value. The D value is the amount of time that you expose the organism to sterilizing by any other method where you've reduced the number of organisms by a factor of 10. So, for example, I just happen to still remember this, even though it was decades ago. Lactobacillus lactis, which will contaminate tomato sauces and tomato paste, has a D value when you've exposed the tomato paste to 240 degrees, it has a D value of 2 minutes. And it says Lactobacillus lactis tends to be found on the skins of tomatoes when they come in from the field. Those tomatoes are washed, usually in a chlorine bath. Then they go through a pulper. They're heated during the pulper. After they go through the pulping, they then go through a mixing process, being heated all the time. Then they get poured into the cans, and then they get cooked. And that's essentially when you're asking to kill the organism at that time. Even though you've reduced the number of organisms on the tomato, you've also been introducing organisms, things that are on the blades and things like that. So it's here that you calculate, you have to know, well, have a good prediction, how many organisms are going into the paste by the time you cook it. That tells you how long you cook it. And we had calculated it such based on sampling at different areas that's needed to cook the paste for 8 minutes. So I said, okay, let's cook it for 10. That way we'll be sure. And so by cooking the paste for 10 minutes, they got a product that still tasted fine. And then it went in the can, and it didn't need to be cooked again because we cooked it for 10 minutes and it was, well, that's a D value. And you'll see D values published essentially in various books. Okay, and so the higher level of microbes, the longer it takes to destroy, and temperature and pH dependent and blah, blah, blah. All right, that's all we have time for today. So we'll finish this on Monday and get into antibiotics. That'll be more directly relevant to your training as nurses. Have a good weekend. This weekend, my science colleagues and I will be conducting a video program that apparently will be used for donors. They'll be shown to donors to raise money for a new science building. So pray for us. I have never been good in front of a camera. So pray that I don't get tongue-tied. And this is why I was Professor Groover in our town in high school, and that's the extent of my acting career, and be very thankful. So just pray for me that I don't get tongue-tied and I don't look like a dork in front of the camera. Well, I will look like a dork in front of the camera, but I don't look like a big dork in front of the camera.